Hey everyone, so I am Steve Rodriguez, and this is our presentation for image acquisition and processing, and then I am with Armando and Fabian. All right, so now we're going to go into the introduction of this lab and the purpose why we did it. So we gained a general knowledge of how to use the webcam with MATLAB and how to change into the different channels to red, green, or blue, and see how much percentage of that is being used with the camera and the picture that was taken. We were able to use MATLAB efficiently to retrieve images from the computer and the computer's webcam and separate the images, as I said, into three different channels. And with this information, we made charts and graphs such as histograms to be able to analyze the data efficiently. Yep, and now onto the materials. The materials that were used for the lab were a webcam, the image acquisition toolbox for MATLAB, which allowed us to take a picture with our MATLAB, and obviously MATLAB on the computer to be able to run the programs.
So for the methods, we created a code that through MATLAB will take a picture of one of us and then process the image. The image will basically the code will divide the picture in three channels: the red, green, and blue. So basically RGB channels, and then we'll show in a grayscale, and we show the content of those channels with a histogram. We formatted the picture to be 500 by 500 in order to better show the content of the picture, so not to make it too big or too small. And then we also did a region of interest. So what we did is we make the code so it can pick a region of interest, and then we'll do the same analysis that we did for the whole picture, but just for that region of interest. In this particular case, we're going to use the forehead of the person taking the picture. So this is an example of what it will look like. We take the picture, it will divide it into three channels: green, red, and blue, with the content shown, and then it will regroup the channels into one showing the original picture to see that we can divide it and then put it together and it will look exactly like the original. So now we're going to show how the code actually works. We were running into some problems when sharing the screen and using the code, so we're going to use an external source, meaning another computer, to run the code on it, and we're going to show it through a camera. So basically, this is how it's going to be. I will show the computer, I will run the code on this computer, and it will come up and show it through the camera. So
could you stop sharing the screen so we could see the screen bigger?
Absolutely.
You could just click it.
Like this?
Stop sharing it. Yep. There you go.
So here we go. This is the new code. And then we're going to click run. I'm going to get a little bit closer to the camera so it can take a picture of my face. And here we see it. So this is the picture that it takes of me. If we look at this, it shows the original picture, three channels with the content is a 500 by 500. And then it shows the original picture regrouped on the bottom. Then if we can take a look at the histograms that it shows, here we have histograms showing the content of the picture in the grayscale. So here we have all the contents for the red, green, and blue channels. Also, if we go back to the original picture, we can select the region of interest, as I said, and we're going to select the forehead. So as soon as we select the forehead, we're going to get exactly the same result. We're going to get another picture just for the region of interest that show the original picture, red, green, and blue channels, and then the recombined image again with the scales, and also the histograms for the region of interest showing the content of the region of interest. So here through this code, we're able to effectively analyze an image and a region of interest of that image itself. Now, Fabian is going to talk about the discussions and the results that we had.
Okay, overall, this lab was very simple, but it was very interesting at the same time because, like Armando said, we're not aware that this was doable on MATLAB. So after we got the code running and we put everything together, and we actually got to see it, it was like pretty interesting. We all had to work together to make it work because it had some small bugs and stuff, but in the end we figured it out. Then we got to selecting the area of interest I was requested, and it worked again. There was some, it avoided all the noise in the data and here in the images I was taking. It was pretty good. Now, in conclusion, the purpose of this lab was to develop an effective code and show images from each of the RGB channels in grayscale with their respective color bar. We just use MATLAB to retrieve images from our computer webcam and separate the image into the 3 different RGB channels. In retrospect, this lab was exponentially easier and straightforward than others since it was a lot less technical. The lab was a success since the team was able to successfully read and analyze colors in images using MATLAB. The lab was not only a good learning opportunity, but it was also entertaining. We enjoyed to be able to see the different colors in the pictures.
I just want to add one little thing at the end, and is that this code that we made also gives us the opportunity to save the picture into the computer, not only just analyze it in MATLAB, but we have the actual picture in the computer that we're going to submit alongside the lab. Thank you.
Thank you. All right.